Okay, so hi, my name is Rob Wimbish. I'm a postdoc in the Omeyer Lab in the Department of Biochemistry and Cell Biology. And today I'm going to be telling you a bit about the work we've been doing in trying to understand how microtubule dynamics can be regulated through a process called biomolecular condensation. Uh-oh. Okay. And so our lab is really interested in this question of how microtubule dynamics can be regulated. And so for those who don't know, the microtubule network is a critical component of the cellular cytoskeleton, and it's really involved in a plethora of processes that are vital for cellular health. And so here I'm highlighting a couple of examples of that. So on the left, you have a cell that's undergoing division. And so during mitosis, the microtubule network forms this football-shaped structure that's ultimately going to segregate the chromosomes into two new daughter cells. So you can see here the microtubules are in magenta, and there's a protein complex called the kinetochore that binds to the chromosomes, and the microtubules are going to physically push and pull on the kinetochores and ultimately divide them into the two new daughter cells. And so another example is here on the right, which is a cell that's undergoing migration. And hopefully what you can see is that as this cell migrates across the field of view, the microtubule network really dynamically changes its shape in order to follow the cell's migration. And we know that microtubules have a lot of interplay with another cytoskeleton component called actin, and that the interplay between these two networks is really important for cell migration. And so in addition to this, there are really a battery of other processes that microtubules are critical for that regulate cellular health and fitness. And so it's really a crucial component for organismal health in general. And so really a hallmark of microtubules in all of these processes within the cell is their ability to stochastically switch between growth and shrinkage. So in order for the microtubule network to have these dynamic structures and to really change its shape over time, the microtubules themselves have to be able to be highly dynamic. So this is an example of that. This is just a look at what individual microtubules within a cell look like. And you can see that indeed they're really rapidly undergoing these switches between growth and shrinkage phases. In fact, the reason for this is because microtubules themselves are highly dynamic polymers. And so microtubules are built from this subunit called tubulin. So there's a protein dimer called tubulin, and tubulin normally is bound to a molecule called GTP, represented as red and green here. And so when tubulin is bound to GTP, these proteins can add to the end of the growing microtubule polymer. But GTP rapidly switches into a molecule called GDP when it undergoes hydrolysis. It's represented in blue in this cartoon. And this is really unstable within the microtubule lattice. So as the microtubule subunits are adding to the end, this GTP is quickly going to hydrolyze. And if the hydrolysis of GTP catches up to the growing end, the microtubule is going to switch from growth to shrinkage. So you can imagine then that the balance between growth and shrinkage of individual microtubules is really a function of how quickly tubulin is adding to the end versus how quickly it's hydrolyzing the GTP into its GDP form. And so this kind of brings us back to our original question of how can the microtubule cytoskeleton undergo these dramatic structural rearrangements in space and time? So again, this is an example of a cell that's transitioning from interphase on the left into mitosis. And you can just appreciate, hopefully, that the interphase microtubule cytoskeleton, it really has to be broken down and completely remade into this, this American football or rugby ball shaped structure that's going to divide the chromosomes. So you can imagine with what I just told you that if each individual microtubule in a cell is undergoing stochastically these switches between growth and shrinkage, a cell would never be able to coordinate such a structural rearrangement in space and time. In other words, if these individual microtubules are kind of randomly growing and shrinking, the cell would never be able to synergistically change the shape of its microtubule network. And so one of the ways to solve this problem is through the addition of proteins that bind to microtubule ends to regulate their dynamics in space and time. And so a class of these proteins are called plus-tip interacting proteins, or plus-tips. And so as the name implies, they bind to the growing plus ends of microtubule where they regulate microtubule dynamics and play a battery of other roles within cells. And so if you express plus tips in cells, this is what they look like. So this is a plus tip that we'll talk about today called N-binding protein 3, or EB3. And so here you're looking at a GFP tagged version of this protein in a cell. And what you can see is that it forms this characteristic comet shape where you're seeing really these streaking comets going through the cytoplasm of the cell. And this is because you're not looking at the microtubules themselves, but you're only looking at the growing ends of the microtubules. And so whenever one of these microtubules switches from growing to shrinkage, this protein actually stops binding to the end and you lose the presence of this comment. We can also look at this by using purified proteins in vitro, which we'll be talking about quite a bit today. And so one of the ways we do this is by purifying EB3 and using purified tubulin and growing microtubules and seeing how it behaves. And we can really recapitulate this activity as you can see here. So here we have a magenta microtubule and EB3 is labeled in light blue. And you can see that anytime this microtubule is growing, you have a really nice signal of the end binding protein on the microtubule end. Whereas anytime the microtubule switches to shrinkage, you lose that signal. And this is maybe a little bit more clearly represented if we make a chymograph of this microtubule. So if you're unfamiliar with chymographs, it's basically just a projection of the one-dimensional position of the microtubule over time. So for example, on the y-axis here, we have the time of the movie, and on the x-axis, we have the position of the microtubule. And so hopefully what you can see from this chymograph is anytime the microtubule is growing, you have a really nice signal of EB3 on its end. And whenever it switches to shrinkage, you lose EB3. So we know that this behavior is existing both in vitro and in cells. And so when we started on this project, we had two main questions about the end binding proteins in particular. And so the main question we wanted to address was, how are these plus-tip networks spatially organized at the microtubule end? And so there's two key properties that are important here. One is that these networks are really highly concentrated. So you can have hundreds of these proteins within a space of, a few tens of nanometers at the microtubule end. Additionally, they're very, very dynamic. So a lot of these proteins turn over on the microtubule end and are replaced by others in a time frame of a second or even less, even hundreds of milliseconds. And so as it turns out, these two properties of being really highly concentrated and really highly dynamic are very typical properties of biomolecules that undergo phase separation or biomolecular condensation. And so phase separation in biology is this process by which biological molecules undergo a somewhat spontaneous partitioning into two distinct phases in a concentration-dependent manner. In other words, as you increase the concentration of these biomolecules, they're eventually going to change into two separate phases, one of which can be represented by liquid-like droplets, and the other of which is more soluble protein. So as it turns out, we've known about phase separation in the context of physics for quite some time. So anyone who likes to add vinegar into oil and eat it with bread knows that when you pour the vinegar into the oil, you see the separation of these two liquids into their own phases. But it's kind of a recently discovered phenomenon in biology, and it's come to light in the past 5 or 6 years that these processes are really critically involved in the regulation of gene expression, especially with RNAs and RNA granules. Phase separation seems to be a very prominent way by which cells can sequester RNAs and spatiotemporally regulate their expression. And so, knowing that the plus-tip network had these properties of being really highly concentrated and highly dynamic, we wanted to look at it through a new lens and ask whether or not phase separation could underlie plus-tip organization. And we were kind of inspired by one of the seminal papers studying one of the earliest discovered plus tips called CLIP170, or cytoplasmic linker protein 170. And so this protein was discovered here at the University of Geneva. And in this paper from the 90s, what they saw is that if they overexpressed this protein, it formed these large patches in the cytoplasm of cells. And so we wanted to revisit this and ask whether or not these patches could actually be biomolecular condensates that formed from really high concentration of CLIP170. And so the work I'm going to tell you about today was a really nice collaboration between myself and Julie Miche, a PhD student in the lab, who did a lot of the droplet assays and the cell studies, and Marie-Claire Velou, who did a lot of the protein purification and cloning. And so we started by revisiting this question about what these patches are when you overexpress CLIP. So we overexpress CLIP in RPE1 cells, and what we could see is that there's really this, canonical comet formation where you see the protein binding to the microtubule plus tips. But in addition, we could see the formation of these patches in the cytoplasm. What we notice is that the patches are actually quite dynamic. So you can see examples of fission where one patch divides and becomes two. And conversely, we can see examples of fusion where two patches can really merge and form one. Additionally, we saw that they're very dynamic by photo bleaching studies. So if we photo bleach half of the condensate, what we could see is that the fluorescent signal really rapidly recovers, indicating that there's a high amount of exchange between the cytoplasmic pool and the droplet pool. So to study whether or not this was due to intrinsic properties of CLIP or whether there were other proteins in the cytoplasm that were influencing these patches, we purified CLIP-170 from insect cells and tried to reconstitute this phase separation in vitro. When we did this, what we saw is that even at nanomolar concentrations, CLIP really readily underwent phase separation, forming these droplets. And we could increase the phase separation of CLIP by increasing the concentration. Additionally, these droplets seem to be highly dynamic. So we could see examples of fusion where, again, two droplets merge to become one. And we saw that there's a rapid recovery after photobleaching, although to a lesser recovery extent of what we observed in cells. We saw that, with a similar half-life, we could recover a lot of the fluorescence of these droplets in vitro. And so we next wanted to ask whether or not other plus tip proteins were involved in this. And so I mentioned to you earlier in binding protein 3, this was the one that we were looking at the chymograph of in the introduction. So in binding protein 3, as it turns out, is an interactor of CLIP, and they have multiple binding sites for each other. So this made it a really interesting candidate since we know that multivalent interactions can really underlie phase separation. Another interesting reason to study EB3 is because CLIP-170 itself doesn't actually bind to microtubules ends. It needs end-binding proteins in order to bridge it there. So for example, if you use an in vitro assay where you have GFP tagged CLIP-170, in the absence of EB3, you don't see any GFP marking the ends of the microtubule, as you can see here. However, if you add EB3 into the reaction, you now have these nice characteristic streaks or comets of CLIP-170 binding only to the growing ends of microtubules. So since we know that these proteins interact and that CLIP needs EB3 to bind to microtubules, we thought it was a good candidate to test for phase separation as well. And so we purified EB3 and we saw that indeed it does undergo droplet formation, although at higher concentrations than CLIP and at much smaller droplets. And when we incubate these proteins together, we see that actually they can synergistically phase separate together, which is to say that if we look at the phase separation capacity of CLIP alone, in addition to that of EB3 alone, it's lower than if we add the proteins together, indicating that they can really form a dense multivalent network that can enhance phase separation beyond the capacity of its individual parts. We then wanted to go a step further and ask whether tubulin was involved in this process. And this question, of course, comes from the fact that the main role of these proteins in cells is to bind to microtubules, right? So it makes sense that maybe tubulin could be somehow partitioning into the droplets that these proteins are forming. So we again stayed in vitro and incubated these reactions now with soluble tubulin. And indeed, what we see is that in the presence of CLIP, tubulin can partition into the CLIP droplets. It also does this into EB3 droplets. And in the presence of both CLIP and EB3, tubulin can additionally partition into these droplets. And it's important to mention here that tubulin alone, even at really high concentration, does not form droplets. So tubulin is not phase separating by itself, but in the presence of droplets formed by EB3 and CLIP, tubulin can then partition into them. So this led us to this idea that tubulin and EB3 and CLIP can kind of coordinate to form these droplets where proteins can partition in and out of them. And we wanted to ask then, what happens when we add these to growing microtubules? So we turn back to this assay that I mentioned earlier, where we grow dynamic microtubules from purified tubulin, and we can add in purified plus tip components and assess how it affects growth. And so this is an example of what that looks like. When we add CLIP-170, EB3, and tubulin together, we get really, really rapid growth of microtubules. And you can also notice that there's really robust formation of these droplets along the microtubules as they grow. These are just some insets to show that. So hopefully you can appreciate that you quickly get the formation of these droplets all along the microtubules that are growing. And these droplets really increase in intensity over time, both for the CLIP-170 and for tubulin, indicating that they're able to pull soluble protein out of the soluble phase and into the dense phase as they're growing. This is just a quantification showing that indeed tubulin does really, really increase in these droplets over time. And so we wanted to assess how this changes microtubule dynamics. So again, I'm just showing you a chymograph here. It looks a little bit different from the one that I showed you in the introduction, just because one, this microtubule doesn't shrink at all. So you can see that the microtubule really undergoes a robust growth phase throughout the entire timing of the movie. In addition, there's a lot of tubulin that's forming droplets all over these microtubules. So this leads to a kind of striped pattern all over as they're growing. So we wanted to measure microtubule dynamics and see how this compares to the presence of non-phase separating CLIP-170. And so we can use this mutant H2 that's actually phase separation deficient, meaning that it does not form these droplets in vitro. And indeed, what we see is that the microtubule dynamics are quite changed. So you can appreciate, hopefully from this chymograph, that there's a really big change. When we actually measure these dynamics, we see that the growth rate doubles in the presence of a phase separation competent CLIP, whereas the catastrophe frequency, which is how often a microtubule switches from growth to shrinkage, is really dramatically reduced, as is the pausing frequency. So what we think is happening here is that when you have a phase separation competent mutant, you get the really robust formation of droplets all along the microtubules, and these droplets are able to recruit more protein and tubulin into them. And this really dramatically increases the growth speeds of these microtubules. So I told you earlier that these proteins are, they're tip binding proteins, right? They bind mostly to the tip of microtubules. So since in this assay, they were binding all over the microtubule, we wanted to understand what would happen if we pushed them specifically to the tip. And so to do this, what we did is we increased the salt concentrations, and by increasing the salt concentrations, we can slightly reduce phase separation, but also push these proteins closer to the end of the microtubules. This is an example of what that looks like. So if we drive proteins to the end of the microtubules, indeed, we see that there's still really robust growth phases. And when we quantify the differences between full length and H2 CLIP, what we can see is that there's still a dramatic difference. So we still get a doubling roughly of the growth rate and a really high increase in rescue frequency, which is how often a microtubule switches from shrinking back to growth. And so this leads us to the idea that when these proteins are concentrated at microtubule ends, they're really robustly able to drive microtubule growth speeds. And so finally, we wanted to turn back to cells and ask whether or not this property holds true in cells. And so these are examples of comets and cells, which I showed you earlier, but maybe you notice that these ones look slightly different. The reason for that is because as this comet grows, you'll notice that it leaves behind remnants of protein behind the leading edge of the comet. These are just some insets of what that looks like. So as the comet is growing, you can see that there's protein remnants that are left behind and that these dissolve over time. And so to us, this looked somewhat reminiscent of what we had observed earlier with the cytoplasmic patches, whereby protein droplets, parts of them could break off and undergo fission. And so we were curious whether or not these remnants that were behind the leading edge of the comet were due to a fission-like process. So we went back and we measured the amount of plus-tip networks that had these remnants behind the leading edge of the comets in cells expressing either a phase separation competent CLIP or H2, which again is this mutant that does not undergo phase separation. And what we saw is that indeed there's a really big reduction in the amount of remnants present when we have this H2 mutant. In other words, for the formation of robust remnants behind the growing microtubule end, you really need a phase separation competent mutant. We also measured endogenous protein using antibodies to get rid of any artifacts that could come with overexpression. And we see that indeed for both endogenous CLIP and endogenous EB3, there's still a really high occurrence of remnants behind the microtubule tip. And often we would see that these remnants actually co-localize with each other if we co-stain cells for these two proteins. So you can see that there are occurrences in which you have high amounts of remnants that really co-localize with each other behind the leading edge of the comet. Finally, we wanted to test what would happen if we could perturb phase separation by using a small molecule called 1,6-hexanediol. So hexanediol is an aliphatic alcohol that's known to perturb weak hydrophobic interactions, and it's been commonly used to reduce phase separation, especially in the RNA biology field. And so what we did is we expressed cherry-tagged EB3, and we treated cells with hexanediol and measured the size of the comets before and after hexanediol treatment. And what we see is that there's a large reduction in the comet size after cells have been treated with hexanediol. In other words, you can really reduce the size of the plus-tip network by treating with this small molecule that's known to inhibit phase separation. Finally, we wanted to measure microtubule dynamics and ask whether or not these proteins had an impact in cells on how microtubules grew. And so we expressed cells, we depleted rather EB3 and CLIP from cells that were expressing GFP tubulin. And what we see is that there's about a 20% reduction in the growth speed when you get rid of EB3 and CLIP, really indicating that these two proteins, which we know undergo phase separation, are really critical for regular growth speeds of the microtubule network. And so just to wrap everything up and conclude, what we showed is that EB3 and CLIP both can undergo phase separation, and during this process, they can condense tubulin into their droplets. If this experiment is done in the presence of microtubule, they condense tubulin on the microtubules, and they can cause really robust growth of these microtubules. And when they're pushed to the end of microtubules, they still cause very, very robust growth, indicating that this activity could be important in cells for their ability to promote growth. And additionally, we showed evidence that the comets in cells have some liquid-like properties. And so with that, I just want to thank everybody who was involved in this work, especially Julie and Marie-Claire in the lab for all of their help with the project, the Access platform for help with imaging the droplet assays, and the Protein platform for help with all of the purification from insect cells. Thank you.
Great. Thank you so much, Robert. Let me just